Good morning, everyone. My name is James Polli. I'm a faculty member at the University of Maryland. And hopefully you have for those of you in the room, and I think we have also people online, there is an agenda. So we're going to start session 1. There's also sessions 2, 3, and 4, as well as some closing remarks. So this session is on advanced manufacturing. I just want to take just a little bit of time just to put some context into this session. So if compared to the other sessions, which are very more clinically oriented, this is less clinically oriented. And in my world, which is a drug world, is often denoted to be non-clinical. It's literally called the non-clinical world. But it's a very big world for those of you unfamiliar with it. It's very big. In my estimation, the non-clinical world is actually bigger than the clinical world. And as we heard yesterday at the summit, this world of development, the sort of non-clinical development phase, as well as manufacturing, is really very, very critical. It's a big part of actually what we expect the FDA to do. I pay a lot of attention to the Office of Pharmaceutical Sciences, as well as the Office of Generic Drugs. So there's lots of folks that participate in this area. And there's, I think, great opportunity for academics to be more involved. We're, I think, in our 13th year, and we love working with the FDA. And one thing I would just add, I was kind of asked this question a little bit earlier this morning. I think one advantage of being able to work with the FDA is it actually does have a real positive impact on faculty who then are able to tune their thinking and gear things that are more application with a regulatory outcome, which is, I think, good for even just the university. I apologize, I should say Bill Bentley. He's not stuck in Hawaii. He's on his way to Clemson, which is a very, very nice place also. But just to give some context about some of our speakers, so the non-clinical world is sort of large and in some ways hard to manage if you're not used to that world. So there's a range of medical products that the FDA regulates: drugs, biologics, cell gene therapy, devices, and we have a little bit of that built into this. So for example, Courtney will be speaking a little bit later. We'll be talking about devices. As a drug person, I think most device people say drugs are complicated. All the drug people say devices are complicated, and she's going to give you a good example of why I think I'm right about that. Matt will be talking about the very important topic of how do we assure quality of these very complex biologicals. And then on top of that, there's always a need for measurement science. Quality has to be measured. And our first speaker, Marc, will be talking about that, about using advanced analytical methods. Marc is Associate Research Professor at the University of Maryland. He's very fond of nuclear NMR, and he'll be talking about that in the context of product quality, applying NMR for product quality. Marc?
Good morning, and thank you, Jim, for this nice introduction. And I'm here representing the School of Pharmacy. And at the same time, I just need to mention that our lab is located physically at IBBR, Institute for Bioscience and Biotechnology Research, which is a joint venture between National Institute of Standards and Technologies together with the University of Maryland, both campuses. And that cooperation provides a lot of impactful results for very broad scientific applications in the pharmaceutical industry, both and everywhere. And today I will be having the privilege to address this audience and give you some interesting results of the application of a fascinating water proton NMR technology for characterization of gene and cell therapy products. And let me briefly outline the most important features of this technology. This technology is very similar to MRI. And what happens there, it's non-destructive, and it uses the water molecules which are abundantly present in the biopharmaceuticals or in the process flow in the manufacturing of biopharmaceuticals. And it uses the water molecules as the signal source. So water interacting with the API of any pharmaceutical product or some ingredients like excipients or adjuvants, is a reporter of the wide variety of different CQAs. And our technology allows you to collect quantitative data non-invasively and in a contact-free, inline real-time mode, both in static and in flow conditions if you want to use it for continuous biomanufacturing applications. And unlike many of us know probably about nuclear magnetic resonance spectroscopy, and you imagine this is some kind of the large cryogenic instrument, several hundred megahertz or gigahertz. No, this technology is implemented using very small benchtop nuclear magnetic resonance machines, low field, and they have a wide bore of this instrument. And indeed, the wide bore of these instruments that you can see on the photos allows you to accommodate the full drug vial in the original container without taking it out. It could be a syringe, it could be an insulin injection pen, it could be a large vial with the cap sealed, everything, and you can do these measurements non-invasively without taking it out, without sampling. And after your inspection, your drug is still usable if it passes the inspection. And inspection in general takes several seconds depending on the concentration of your API, depending on the size of the bottle, from less than 1 minute. The most important global parameter is the relaxation rate of water. And you can take it within several seconds easily. So let me briefly outline what applications we already have been explored using this technology. It could be used almost in every step of the life cycle of a drug product, starting from the formulation, where you can monitor the drug product stability or drug substance integrity, going to the manufacturing, where you can monitor the concentration of your drug, presence of aggregates, and other critical process parameters. And at the same time, it's probably the single technology right now, as far as I know, which is capable to monitor the drug product quality integrity at the point of care and point of release, non-invasively, so the drug can be used again. We have already demonstrated and validated our technology using a lot of different orthogonal techniques for different CQAs. For example, using water NMR, we demonstrated that we can monitor the content uniformity of many monoclonal antibody formulations. For aluminum adjuvanted vaccines, insulin pen formation of the complexes between adjuvant and antigen in the vaccines. We also use it to monitor the stability of drug products, monitor aggregation formation, agglomeration, and clustering in different MAP products, aluminum adjuvanted vaccines, insulin pens, and nanoparticle formulations. And most importantly, quite recently, we have explored the use it for the safety of drug products, demonstrating that water NMR could be also used to detect the counterfeit vaccines and biotherapeutic. But let me now show you some very recent results that we have obtained in application of our technology to the characterization of gene and cell therapy product. So I might be preaching to the choir here, telling that the understanding of the empty-full ratio in the viral capsids is one of the most important CQAs of the gene therapy products. Here in this particular experiment, we had two samples with the 100% full and 100% empty vaccines. This has been validated by using standard AUC technologies and UV absorption into 60 to 80 nanometers. But when we did the experiments with these two samples, you can see that water NMR global parameter, or relaxation rate of water, could be easily discerning between these two formulations. They have identical concentration of viral genomes per milliliter, but the difference between is significant, and it's very easy to discern between these two formulations. Going further, we use these two samples to mix them in a different proportions to create the dilution series, where we definitely know how many of the full capsids and how many of the empty capsids are present. And amazingly, from this linear dependence that you can see, we can easily define the empty to full ratio with the precision between 1 and 3%, which is not even attainable by the standard technologies right now. Moreover, this technology, once you have created the calibration curve here, you don't really need to do any other characterization or validations. Water relaxation will immediately tell you how many full and how many empty capsids you have in the sample. And of course, in thinking about that, we also will looking at the possibility to use our technology to count the cells in a different kind of cell culture. How many viable cells you have there? What's the cell counts? This idea was bugging us, our lab, for quite a long time. But to do that experiments, we really needed a very nice partner who have been doing these experiments for us for validation of this technology using the cell counters. And because we, as the pharmaceutical scientists and mostly analytical instrumentation users and developers, have a little experience in cell studies and cell formulations and preparations. And we found these colleagues in the NIST, because NIST at this time was developing the CHO cells, NIST CHO cells reference materials, and they were eager to collaborate with us. And so what we did here, we explored the use of the water NMR technology to monitor the cell counts and cell viability in the same culture when it is fresh and 97% viable cells, and 1 week after that. From what you can see here, this linear dependence allowed you to estimate the cell counts with a coefficient of erasure of 0.9%, which is significantly lower than any cell counting technology. At the same time, if you see here, between the variation, the difference between relaxation rates observed for the 70% viable cell culture and 97% viable cell culture is so significant that you can have a 3 to 5% accuracy to define the viable cell percentage in these cultures. And most importantly, we also looked at whether this technology is reproducible. So what we did, we created 2 identical cell cultures under the same conditions, same cell types, same growing, but we created them 1 month apart. And when we measured these 2 fresh cultures, we observed that we have almost identical results for relaxation rate. That tells you that if you have the same type of the cells, same conditions of their growing, time and medium and concentration of CO2 and temperature and shaking rate, you will obtain the identical results from the relaxation rate, and that allows you to reproducibly do these measurements without actually calibrating anything. You need to do it only once for your culture. We also have tried to find out whether this technology allows you to observe very old cell culture. So we have created 1 culture and then compared these results with the same culture when it was 1 month old, where only 11% of the viable cells were there. So as a result, you can see that there is also a great distinction between these 2 when you do these measurements. At the same time, the differences here is a little bit because we use the conditioned media, not the fresh propagation media, to create these linear dependencies, but you see that there is a great difference between the 2. So to summarize, we can see from this experiment that water NMR technology could be applied to analysis of gene therapy products and to find out the empty full capsid ratio. Also, it's a simple rapid data collection on the cell counts and cell viability. And it also might be used to monitor the growth and aging of any cell culture. What is very important is that water NMR technology does not compromise the integrity of your sample. And after the measurements, I can give it back to the experimenter, back to the patient, back to the doctor, because it's like MRI, it's not changing anything. And it's very important, for example, for very precious samples like gene therapy products or CAR T-cell therapies, because it allows you to save precious sample for future use, and you give you an accurate count of everything. And application might be applied to any stage, from process monitoring for QC and even in the point of care for the individual patients. And here on this, if you go to this QR code, you will get all our selected publications from our group. With that, I need to thank my friends and colleagues from the University of Maryland School of Pharmacy, Professor Bruce Yu and Dr. Katie Briggs, my collaborators from NIST and Institute for Biotechnology Bioscience Research, Dr. Brad Odell, Dr. Yannis Karagiorgis, and Dr. Tzvi Kelman, and also Dr. Mike Jones from Pfizer and I need to thank Pfizer and the CERSI for this financial support, and I will be happy to talk after that.
Thanks, Mark.
Yeah.
Thanks, Mark.
It's after that, right?
Yep. Okay. Yeah, we'll have a Q&A panel, a brief Q&A panel before break. Our next speaker is Courtney Moran from Johns Hopkins Applied Physics Laboratory. Just met Courtney this morning, very enthusiastic. I'm not sure I can say the word prosthetic clinician, okay, but with interest in outcomes research, but also the design of these devices or systems of devices. Not sure what the right how to exactly characterize those end products.
Thank you. Good morning. How's everyone doing this morning? My name is Courtney Moran. I have a master's degree in biomedical engineering specific to implantable prosthetics, and I'm also a prosthetic clinician. And today I'm going to be presenting our preliminary outcomes of the design of a persistent inline sensor for real-world loading characterization of osseo-attached prosthetics and the osseointegrated implants they are connected to. So our CERSI collaborators, so the ones on the team members are myself, Jonathan Thornhill, Luke Osborne, Ajira Wormley, and Jill Curran. Our FDA technical collaborators or technical support team was Kim Conson, Snehal Shettia, Kinera Chadha, and Ruiz Lee. And we did have in-kind collaborators from Uniformed Services, University of the Health Sciences, and Walter Reed, Michelle Nordstrom, who is the occupational therapy director of the DoD Osseointegration Program. And before I go any further, if anyone's not familiar, osseointegration is a transcutaneous implant that goes into the bone, through the skin, and limb prosthetics are attached to that. And so the FDA regulates those implants, and then prosthetic devices, depending on their level of complexity, are also regulated, but you have different classes of devices attaching to one another. So understanding how those are used outside of a controlled clinical environment is our focus on this development effort. This project ran from September 1st, 2022 to August of 2023. And so the challenges that we're facing is that there are load sensors that can be used with any prosthetics to determine like gait kinematics and loading characteristics, but they're excellent. For many amputees, they're too heavy and bulky to even accurately quantify loads of movement because they interrupt the kinematics and oftentimes require removal because of their height and size of some of the safety hardware that goes along with osseo-integrated implants. Patients are reporting, however, that the loads that the activities they do far exceed some of the safe loading constraints that we have put on them as a user, and that they're doing so safely. And so the interest of the FDA was to really understand what it looks like in real world and to quantify those loads so that the appropriate regulatory spectrum can be applied to these implants. But without understanding that outside of the controlled clinical environment, that's very hard to do. And so we are working to develop an implant that's much simpler, can be used with all level amputees, and provides and has the data security to ultimately go out and be part of an overall prosthetic device. So This focus was the initial development of the physical system and validation of the loading in 2 directions. But basically, our focus was on the CDRH priority area of collection of real-world evidence, and then also the increased post-market surveillance. So understanding that real post-market surveillance once that goes out. So we did also apply some advanced material capability. We vetted multiple designs for the physical interface using FEM analysis of our materials and design characterizations. And then we worked with Michelle Nordstrom. I'm also a clinician, and we have some clinical research history at APL with the modular prosthetic limb that allowed us to have measured values for these upper limb bone-attached amputation use characteristics. And so we established our user-driven requirements with Michelle and myself. And then our goal was to fabricate and test at least 1 low-profile sensing design prototype in at least 2 of the loading directions of interest. And ultimately, we were able to do that, and I'll show you some of that data. It weighed only 3 grams. Now, that doesn't include boards or any power, but it's 161 grams lighter than our target, so it gives us a lot of room to work and maintain that. And the total size was 4.5 centimeters by 2.5 centimeters, which was 1.5 and 0.5, relatively speaking, smaller than our target. So we're hitting that target for our just general form factor. So this is what that looked like. It looks really basic. One of the major constraints for this development was making sure that it was available to be used with any prosthetic componentry that might be attached, and that it interfaced with any of the safety hardware that these implant designers have as part of their connection componentry. And one of the basic connection layouts is this 4-hole pattern, which is consistent across commercial off-the-shelf prosthetic designs. Because we are not applying, and because we would have to actually interface with each manufacturer of the different implants, we are actually sensorizing a device that goes just below that attachment. So we are interpolating the loads that would be experienced. And so we have to calculate the drop-down distance, but we wanted to make sure we were seeing the sensitivity and specificity around the collar of those 2 plates that align with what we know to be the case of direct measurement at the implant. So we were able to get the size and weight and integrate load sensors in 2 axes of interest. We did bench testing of this system, and the blue boxes here highlight the Whetstone Bridge sensor configuration that correlates with each axis. By tying the different sensors together, we're able to isolate the different loading characteristics and measure them over increasing loads. These are extremely small sensors, so one of the biggest challenges was our amplification and signal-to-noise ratio dampening. We ultimately were able to get our torsional loading and bending loading to be extremely consistent all the way up to the max loads that we had seen when we measured upper limb use characteristics with the modular prosthetic limb. So that limb was a research effort out of the DARPA Advanced Revolutionizing Prosthetics program. That limb is censored in of itself, but it's not a commercial off-the-shelf device. So we had some historical data that we were looking to match with a commercial off-the-shelf device. So when I refer to that, that's what I'm talking about. So we were able to accurately detect those loads in each direction. This is the long-term vision. So up in the top left, that is an example of the research and the setup that we did with the modular prosthetic limb with a bone-anchored or osseointegrated attachment. Originally, that was not the case. We adapted the prosthetic limb to go onto our clinical research participant who had osseointegration, and then utilized this onboard sensors as well as sensors directly applied to the implant abutment to get some loading characteristics. That is what informed our design, and ultimately what we're looking to do is have a closed loop system. It does not need to be complex, it needs to be accurate. At any given time, we need to know what the maximum load is in any given axis that a user is experiencing. It would then alert the user to the loads that exceed the guidance and provide a user interface that would allow them to provide contextual information as to what they were doing at that time. So one of the great things that we have access to at APL is we have a lot of supporting research and research capability. We were able to leverage our collaboration with JHU. So we are a university-affiliated research center of JHU. So we're separate, but we do all kinds of collaboration. So I was lucky to be representing the JHU CERSI on this. And we actually were able to work with the JHU undergrad engineering team as their capstone project. They were looking at some of the other ideas we had for prototypes. So in parallel, they were able to do that to kind of force multiply our effort here. In addition, another JHU team is collaborating with University of Louisville, and they just published information on a spinal stimulator that has a user interface. It so happens that that user interface will take any raw data and can be reconfigured. So that allows us for our holistic kind of end vision to already have that piece in place by leveraging that existing research and also ensuring that we have the data security that's lacking from these commercial systems. And so, we're really lucky to have that, and we see that our next steps would be to develop the software to go on our implant and refine the implant user interface and add tension and compression, the third degree of loading of interest, and then validate all of that, and then incorporate with these other elements for that closed loop system. Over. Thank you so much to FDA and JHU collaborators for helping us with this. Really, really could not have done it without you.
Thanks, Courtney. And our last speaker is Matthew Porteus. He's professor of pediatrics and stem cell transplantation here at Stanford. Beautiful campus. I think we all met you yesterday. Is that right, Matthew? Okay. And Matt has lots of interests, including genome editing to cure kids of disease.
Thank you. All right. Thank you all for being here, and thank you for the invitation to be able to present our CERSI-funded project, which is related to helping establish the scientific data for what should be required for a CRISPR-based genome-edited drug before it enters clinical trials. I'm fortunate enough to be involved with several companies, but this was a collaborative work between my academic lab at Stanford and the FDA through the CERSI-funded program. I'm not going to go through this, but just to highlight that cell-based medicines and gene-based medicines and microbiome-based medicines, which all can be categorized as living drugs, because instead of degrading over time, they have the potential to expand and interact and be durable over time, really give an entirely different PK and PD compared to some of these prior modalities, which is, of course, not to underestimate the bread and butter effectiveness of enzymes and small molecules and biologics, and especially vaccines. We probably wouldn't be here today without the innovations around vaccines that have occurred in the last few years. So my lab is focused on using genome editing as a way of changing the DNA sequence of cells as a way of creating a cell-based drug. And we especially use what might be considered vanilla genome editing, which is to design a CRISPR-Cas9 to make a double-stranded break exactly where we want, and then ask the cell to repair the break. I'll go forward. Repair the break using homologous recombination, which allows us to create single nucleotide changes up to several kilobases of DNA. Now, the concern is the CRISPR-Cas9 system and the guide RNA is about its specificity. I'll just point out that this genome editing using a double-stranded break is actually how nature has chosen to do most of its genome editing in meiotic recombination using the SPO11 endonuclease. But more I think relevantly is that the RAG1, RAG2 endonuclease complex creates double-stranded breaks that's either repaired by non-homologous end joining in mammals to create our immune system diversity, or actually in birds and other mammals, the double-stranded break is repaired by homologous recombination using pseudogenes. So when we're tapping in, while we're using double-stranded breaks to stimulate genome editing, we're tapping into a biologic process that nature has also used. I'll also point out that the genome is not a pure entity. It is changing all the time. In fact, every cell division results in multiple tens, if not hundreds, of nucleotide changes to the genome. And what I list here is 2 papers from Peter Campbell's group at the Sanger, really documenting the hundreds of mutations, if not thousands of mutations, that occur in hematopoietic stem and progenitor cells during the lifetime of a human being. Again, pointing out that we're not talking about some sort of pure entity. I'll also remind you all is that every time you get on a flight and you spend an hour on your flight, cosmic radiation has created 5 billion new double-stranded breaks in your body. So maybe think carefully about how often you want to spend time at 36,000 feet. That being said, some of you are probably in the air thousands, hundreds of hours a year, and the risk of cancer is quite low, which is all to say that double-stranded breaks per se, while having potential deleterious effects, our cells are well handled to deal with them because we deal with so many of them every day. So our system is one where we engineer cells outside the body. We purify our cell of interest, and I'll show you that our cell type of most interest is the hematopoietic stem cell, the cell that gives rise to all the blood cells in the body for the lifetime of a person. We put the cells into cell cycle because that is when the homologous recombination process is most active. We deliver our CRISPR-Cas9 nuclease as a purified protein complex to a purified guide RNA and delivered via electroporation. And then we deliver the donor for the homologous recombination on a non-integrating virus, AAV6. We saw some NMR analysis of how you can assess the purity or the purity of the full versus empty capsids. And using this system, we can get up to 50 to 80 to even 100% now HDR in cell types of interest. So the question then is, how do you make sure that the double-stranded break that you're creating at your on-target site, you're not seeing inadvertent double-stranded breaks at off-target sites that might cause problems? So the first step in that process is actually to use simple computational tools and select your guide RNAs such that they don't have obvious homologous sequences across the genome. And that really quickly eliminates your so-called bad guides. The second step, and I'll show you the data to support that, is since the discovery of the original version of strep pyogenes Cas9, there have now been engineered versions that have increased specificity. Now, some of these versions have increased specificity at the cost of on-target activity, which for a therapeutic program is a bargain that I'm not willing to make. But other versions of these engineered higher fidelity Cas9s have high on-target or identical on-target activity with substantially improved specificity. I'll show you the data on the version from IDT, but this isn't, well, I'm a big fan of IDT. We've collaborated with them broadly, but there are other companies that sell versions of high-fidelity Cas9s. Now, recently, the FDA put out a guidance on the sets of safety data they would like to see in an IND application. And honestly, it's not, I don't know what the right analogy here is. It's not rocket science, it's not brain surgery. The categories that they're asking for are I believe, are quite reasonable. And I put them into 3 buckets. The first is to ask, are you creating mutations at other sites in the genome? And please tell us how you identify which sites you've looked at and how you quantified whether those sites have acquired new mutations or not. Again, the FDA has not said that the threshold is 0. They said, please measure it and tell us what that number is. The second bucket is, well, we want to know whether your engineering process has created structural variations in the genome. And I would say that there's still need for probably better assays here, although Cas-seq is a newer assay that I think is becoming more and more accessible. And then finally, to complement these genomic approaches is they would like you to show evidence of the the functional toxicity of your process. So how do the cells behave, whether it's by simple cell counts and viabilities, but also with functional assays, and whether they have, by the engineering process writ large, have you created a product that might lend itself to becoming a tumor? All of these have the issue of what's the limits of detection. If you're infusing a cell product that has 1 billion cells in it and your limit of detection is only 0.1%, you of course could be missing a large number of cells that have an off-target or an event that you just can't measure. That is the limit of current technology. So I'm going to describe to you 2 studies that we did. The first was not the CERSI-funded study. This was a collaboration with Illumina, who had developed a panel of cancer-associated genes called their TSO 500 panel. They paid for the sequencing, we made the cells. And what they did is they paid for very, very deep sequencing of these 500 cancer-associated genes from cells that have been treated with a CRISPR-Cas9 RNP, and to see whether our editing process in general were creating any mutations in these cancer-associated genes. And if I go back, the answer was no, so it's really hard to show no. But what was also very interesting is we chose a guide RNA that targets a gene called ZFPM2. I have no idea what that is, but we chose it because we knew it had an off-target in the panel, so it was our positive control. And what you can see in the top left in panel A is that in donors 2 and 3, we saw that there was a measurable frequency between 10 and 25% of indels at the off-target site in the cancer-associated gene EZH2. In donor 1, however, we saw no evidence of off-target indels, even though the sequencing depth was equivalent to donors 2 and 3. And so then Kyle, who's leading the study, we pondered this, and he actually sequenced the target site from donor 1, and what you can, well, it's hard to see because I don't have a pointer, but donor 1 on the lower right actually has a single polymorphism in the guide RNA site that abrogates the ability of that guide RNA to cut that gene, showing that these guide RNAs in certain circumstances have basically a single polymorphism can abrogate cutting, I think pointing out the specificity of the system. This is using the IDT high fidelity guide. Now moving on to the CERSI funded project, this again was developed, this was done in collaboration with IDT, and the idea here was, well, there's a lot of tools out there that people have proposed to identify potential off-target sites. And some of these tools are computationally based or bioinformatics based, and some of these are biologically based either on in vitro assays or in vivo cell-based assays. And so we wanted to explore these different assays and ask what were the sensitivity and the positive predictive value of each of these assays. So we identified a set of 11 different guide RNAs, identified a set of potential target sites of 200 for each potential guide RNA. This 200-site panel was developed based on computational tools, and the known biologic assays had been used for those tools, and then done amplicon-based sequencing to measure whether off-target indels had been created. And what we found is out of the 2,000 potential off-target sites, we only found using RMP delivery in hematopoietic stem cells with intact DNA repair pathways, 11 instances of a measurable off-target indel. And what this slide shows is that when you use the high-fidelity Cas9, you reduce the off-target indel frequency either from detectable to undetectable, as you can see for off-target 3 and 4 for the AAVS1 guide, or from a level of, say, 38% at the HBB guide to something around 1%. So using this high-fidelity Cas9 allowed you to reduce the number of detectable off-targets significantly and the level of indels at any off-target significantly as well. So again, this is looking at the profile of where these off-targets are, and you can see, again, just a small number of sites that actually have measurable off-target indels using the high-fidelity Cas9. They map mostly to non-exonic sites. One of the 11 was in a exonic site. So of the 2,000 other sites, we would call them false positives because we were unable to detect them. False positives only because we couldn't detect them. There was a correlation between where the mismatches were as it relates to the guide RNA position. I'm not going to go through this except as it leads to a conclusion. And what we found is that the biologic methods shown here on the right-hand side as change-seq, circle-seq, discover-seq, guide-seq, site-seq, were less broadly used because they're more complicated assays, whereas the left-hand side, 3 of many, many bioinformatic programs, we could apply to all of the guide sites. And we found that the COSMET system identified all of the bona fide off-target sites when using the high-fidelity Cas9, and that the biologic methods did not add any novel off-target sites that were not identified by a computational method. And so if we look at specificity, sensitivity and positive predictive value, one can see that essentially all of the methods have a very poor positive predictive value because the system is overall quite specific. And the COSMET system was unique in giving a score to the predicted probability of there being an off-target indel. And what we found using the high-fidelity Cas9, with both, is that if the score of the off-target site was less than 10, and this is a pretty arbitrary score, it was only when the score was less than 10 that we see detectable off-targets, but you can still see that the COSMET score or sites with scores less than 10 still were more likely than not to have measurable off-targets, although that was the bucket where bona fide off-target sites were identified. So we now use the cutoff of 10 using the COSMET algorithm as a way of, when we're screening guide RNAs for their off-target potential as the target sites we screen. So in conclusion, I think using thoughtful methods, you can get very high specificity of CRISPR-based genome editing. It's important, I think, to develop accessible, reliable, and reproducible assays that can be used broadly. I think one of the tensions between those of us in academia and those of us who also play a role in translation is in academia, you get rewarded for developing complex, only I can do it assays, whereas what we really want is assays that somebody anywhere in the world could perform in a reliable and reproducible way, and I think we need to continually come to an agreement about that. I think there is evidence now that most of these computational methods were developed 3 or 4 or 5 years ago, and I think there's now bigger data sets out there, and of course with machine learning and artificial intelligence, there's the possibility now to create programs that maintain sensitivity but reduce false positives. Of course, these do require large data sets, and the question is who has big enough data sets. And again, we need to have assays that assess for not just off-target indels, but overall the genomic integrity of a cell. So thank you again. I get to work with a great group of people in my lab, my clinical collaborators. The idea that the working with collaborators around the world, both in academics and in the private sector, has been fantastically useful for us. And I'll just conclude with a picture of some of the people in my lab. I'm proud of all the different faces and looks and that. And I have a world calendar showing up via Amazon tomorrow on Wednesday, and we'll start to document where everyone in the lab came from. So thank you very much. I think we're done with this, and we'll have a panel for questions.
Marc and Courtney.
Okay, so while our panelists are setting down, I was thinking about how, you know, we're coming together with these sessions where it seems like maybe when you go to a scientific conference, you're usually in a session with other people who are presenting similar work than you, and you're kind of building off on top of those foundational elements. And here, and I think throughout the day, you'll hear from speakers who might not be working directly. You know, there will be some elements that are connecting your work. And I think it'll be a unique opportunity for you to see work you might not have usually had the opportunity to be exposed to and colleagues to meet. And so again, thanks to our CERSI program directors for being brave and embracing this inaugural day of figuring out a way that we can bring all of our scientific PIs together. So I just wanted to make sure that was kind of reiterated as our goal for today. So thanks for working along this experiment with us.
All right, yep.
Yeah.
So in that vein, I'd actually like to, I'm going to ask the first question if that's okay, but please let me know if you have a question. We do have only a couple minutes, and I'll actually direct it towards Matt, who spoke yesterday. You were the one who wasn't like the other, if I remember, yes. In the CMC section. And one of the themes there at that session was platforms, right? The idea of having a simple platform, we all work together to make manufacturing occur faster. I have to say that I'm a little skeptical of that, and I don't know your area. So my question is, you know, to what extent are platforms available to make the therapies that you're striving for actually doable? Because I do, you know, one term that came up yesterday, right, was corporate confidential information, and I think there's a lot of that in the development space, measurement space, spec space, design space. So I'm just curious about, for some of these advanced products, how is it?
Yeah. Will you turn on the mic in the back? Or I can just talk loudly. So I think that there is the opportunity for platforms. I think Laura Esserman yesterday commented about clinical trial platforms complementing manufacturing platforms. I'll add in, I think there's possibility for analytic platforms that can be used across, say, manufacturing platforms. I think Rob Califf also made the comment about why he didn't understand why supply chain was necessarily a corporate secret. I would say along the same lines, why do safety analytics have to be a corporate secret? I will give a shout out to the New England Journal of Medicine for requiring the publishing of INDs as part of appendices when they publish a first-in-human CRISPR-based genome editing. Of course, it's redacted for some information, but it does give you a sense of what they did. So I think there is a possibility here. I do think the challenge is that the FDA only gives guidance, and then we have to respond to the guidance. They can't mandate what we do or don't. But hopefully someday we can come to a platform of what a safety package for a CRISPR-based nuclease would look like. I was actually talking to somebody last night at JP Morgan that I love it if a CRO just said, I have a drug master file for a safety package. Give me that and I'll do A, B, C, and D. We'll present the data that's in an IND compatible format and maybe one day a BLA compatible format and present it just as, say, when I order a reagent like a viral vector, my manufacturing CMO gives me a drug master file. So I think there's a possibility.
Okay, all
Yeah.
right. Glad to hear that has possibility with some of the advanced products that you're working on, because I'm kind of skeptical, I have to
It's
say.
going to take time. It's not
Okay,
obvious.
okay. Kathy?
This unfortunately is for Matt. Yeah, so Matt, yeah, sorry. FDA gave us their guidance in which they're asking whether you're creating mutations, structural variations, and functional viability. I think one of the things I would be concerned of if I were a patient is that somebody disrupted a tumor suppressor gene.
Right.
Or so is there is there a feeling to you that they should add criteria like, tumor or activated oncogene, did
Yeah.
something like that? Yeah.
And that's, sorry I went through it. That's why we did the TSO 500 panel, because instead of looking across the entire genome at, say, minimal depth, could we look at the most important regions of the genome as identified by this TSO 500 panel? One could argue whether those 500 genes are the most important genes or not. But let's look with a light at those regions because they're the most important regions. So I think moving forward, we will make that part of our package when we present to the FDA. Again, I appreciate the guidance because it was general and they weren't too prescriptive because this field is so new. If they were so prescriptive early on, I think it would lock things in, and I think in 5 years we might be doing a whole different set of studies than we're doing now.
I know, but it worries me because somebody else may not test those tumors as a patient, if I were a patient. So, yeah.
And that goes to, the risk-benefit analysis between the sponsor and the FDA is have they, for the severity of this disease, have they done an appropriate amount of safety analysis, whether it's directly looking at cancer-associated genes or not. I do have to say, sorry, diving deep in, October 31st, Halloween of last year was when the FDA advisory committee for the CRISPR-Vertex exa-cel product met, and a non-voting committee, which we talked about the pros and cons of yesterday, but they still made it very clear that they thought the theoretic risk did not outweigh the real risks and benefits that have been demonstrated. And I was pleased to hear that that was the conclusion, because we could have easily gone down a rabbit hole of pursuing theoretic risk. So it's a balance. Yeah.
Okay, with the time, I think we're going to have to stop taking questions.
Sorry
We have
about
time
that.
for a short break. We'll be back at 10:30, but much thanks
Yeah.
for our presenters to give us some updates about what's going on at some of the CERSIs. Thank you. See you back in 10 minutes.